Welcome. In this explainer, we're going to do something a little different. We're not just answering a question, we're trying to get right inside an examiner's head. We're going to take a real AQA GCSE Biology exam question and break it down step by step using the official mark scheme. So let's dive right in.
So, here's the big question for you. Could you get all 13 marks available on this one? By the end of this, you are going to know exactly what an examiner is looking for, you'll know the common traps to avoid, and you'll see every single correct answer they are told to accept. Let's get started.
All right, first up, we have a question about the practical side of things. This is a 2-mark question, and it's all about how you'd prepare a slide to look at under a microscope. Let's see what the question actually is.
Okay, so here it is. You can see the image there. A student is getting a slide ready, and the question is pretty straightforward. Name 2 pieces of lab equipment they could have used. There are 2 marks up for grabs here, 1 for each correct piece of equipment. What would you write down?
Now check this out, because this is the really interesting part. Look at the sheer number of possible answers. This is the complete list from the official mark scheme. You could have said microscope slide, cover slip, dye, a mounted needle, a pipette, a scalpel, or forceps. You only need to pick 2 from this entire list to get the full 2 marks.
But this is where we get the real inside scoop. The examiner is also given these specific notes. They can allow you to name a specific dye, like methylene blue, or even mention using a swab. But they are told to ignore an answer like water, which is kind of irrelevant, or knife, which is way too vague. The big takeaway here? Precision really matters.
Okay, let's move on to part 2, where we're going to focus on the microscope itself. We've got 3 quick-fire 1-mark questions coming right up. Okay, question 01.2. It's pointing right to the top of the microscope. This is a super simple identification question. What is part A called?
The correct answer is, of course, eyepiece, or you could just get away with saying lens. But, and this is so important, look at that examiner's note. Do not accept objective lens. This is such a classic mistake. Mixing up the eyepiece lens with the objective lenses down below will instantly lose you the mark.
All right, next up is part B, that big knob on the side of the microscope. The question is asking for its function. Again, just 1 mark up for grabs. So, the mark scheme actually gives you a few different ways to say this and still get the mark. You could go with to focus the image, to make the image clearer, or, if you want to get a bit more technical, to improve resolution. Any of those will do the trick.
And here are those secret examiner's notes again. If you just say it moves the stage up and down, well, that gets ignored. You're just describing what it does, not its function. And here's the big one. Any reference to magnification is a definite do not accept. Focusing and magnifying are 2 totally different things, and the examiner is specifically checking if you know the difference.
Okay, this next one is all about practical problem solving. Imagine this. The student looks through the eyepiece and sees absolutely nothing. So why? What's one possible reason? And wow, look at this list of possibilities. The mark scheme accepts a ton of common mistakes. Maybe there were no cells to begin with. Maybe the slide was in the wrong spot, the lens wasn't clicked into place, or maybe it was just totally out of focus. Any one of these practical, real-world answers gets you the point. And it doesn't stop there. The examiner is also told to allow answers like the light being off, or, and this is a common one, the student just forgot to stain the cells, so they're basically invisible. It just goes to show, thinking logically about what could actually go wrong is exactly what they're looking for. Okay, deep breaths, everyone. It's time for the main event of question 01, the big one, the extended response worth a whopping 6 marks. With these questions, it is all about how you structure your answer. So, here's the task. Compare the structure of a red blood cell with the structure of a plant cell. That keyword, compare, is everything. It means you have to talk about what's the same and what's different. You can't just list a bunch of features of one and then the other. So, let's break this down. The mark scheme is basically our cheat sheet here. For the differences, you could talk about the nucleus, or the lack of one, the cell wall, the shape, that biconcave disc versus all sorts of plant cell shapes, hemoglobin, chloroplasts, a permanent vacuole. See how every single point is a direct, this has it, that doesn't comparison?
But wait, don't forget the similarities. For a top-tier answer, you've got to mention them. Both have cytoplasm, right? Both have a cell membrane. And hey, here's a clever one. You could even get a mark for saying they both have pigments, even though they're totally different ones. Hemoglobin in one, chlorophyll in the other.
Now this is the secret sauce for getting into that top mark band. A level 2 answer, that's 4, 5, or 6 marks, requires you to explicitly compare both cells throughout. I'm talking about using words like whereas, however, or similarly to link your points together. It's not a list, it's a comparison. All right, we are on the home stretch. The very last part of question 01 is a quick 2-mark explanation. This is where we apply what we know about cell structure to a totally new situation.
So the question lays out a classic biology scenario. You take a red blood cell and a plant cell and you pop them into a beaker of pure water. What happens? The red blood cell bursts, but the plant cell is fine. The question is, why? Explain it for 2 marks.
And here it is, the perfect 2-mark answer, straight from the people who write the exams. Point number 1, you've got to say that water enters the cells by osmosis. That's your first mark. And for the second mark, it all comes down to that key difference. The plant cell has a strong cell wall, and that's what stops it from bursting. Boom, 2 marks. Simple as that.
So let's just step back and look at what we've done. In just one single question, we've tackled practical microscope skills, we've done a deep dive comparison of animal and plant cells, and we've covered osmosis. And by peeling back the curtain on the mark scheme, we now know exactly how to nail every single one of those marks.
We've seen exactly what an examiner wants to read, right? We know the specific phrases, the classic traps to avoid, and the level of detail you need for those big 6 markers. So the real question now is for you. Now that you know how an examiner thinks, how are you going to use that to completely supercharge your revision from this point on?